In the last video, we walked through each of these key players of DNA replication. And so in this video, we're going to look at some animated slides to try and bring these abstract ideas to life. So we talked about how in the cell, DNA replication begins at the origin of replication. And it's at those origins where the DNA starts to separate from one another, forming a replication bubble. And in that replication bubble, the new strands of DNA get created bidirectionally. And that's just a time-saving tactic because DNA replication happens really quickly in the cell. Remember, bacteria, they completely divide within 20 to 40 minutes. So that means they have to copy their DNA and kind of elongate and eventually split off into two cells in a matter of minutes, okay? So by having the new strand of nucleotides be created bidirectionally, it's just making sure that all of these important processes happen in a timely fashion. So because of my artistic limitations, remember I am going to be showing you this process just looking at a replication fork, which is half of a replication bubble. So notice, y'all, I'm showing the directionality of these two strands. These are my two parent strands. And you'll notice here, the DNA is closed. That means it is not ready to be used as a template, okay? These strands right here are open. That means the cell can start building the new strand of nucleotides, okay, for each of these. That's what we're going to go through now, okay? So just to remind you, there were 3 enzymes that work at the replication fork. They were the enzymes responsible for getting this DNA separated, okay? So helicase would be working right here at the kind of where the fork is open and closed, because helicase is responsible for breaking these hydrogen bonds and allowing these strands to separate. Then single-stranding binding proteins will bind to these single strands and prevent them from closing again. And then topoisomerase would be somewhere down here in the closed region, making sure that this DNA doesn't get wound up too tightly, okay? Because if it does get wound up too tightly, it can break, all right? So those 3 key players are all responsible for working near the actual fork where the DNA is starting to be opened up. So let's focus on the other key players now. So remember, I said the main player is DNA polymerase 3. But DNA polymerase 3 cannot start from scratch. It needs help to get going. And remember, it's Primase that offers that help. So Primase comes through and lays down a few RNA nucleotides. This is going to allow DNA polymerase 3 to come in and add the DNA nucleotide. So we're now officially starting to create this anti-parallel and complementary strand of nucleotides, okay? So notice, y'all, this parent strand, the orange one, is running 5' to 3'. And notice the directionality of the primer here, which would also be the same for this piece of DNA. Notice it's anti-parallel, okay? Do you see that? See how the primer has a 5' end on the right, which is anti-parallel to that 3' parent strand? Okay, and that is important because remember, DNA polymerase 3 had 2 requirements, okay? Remember, it's picky. And so the first one was it needed help getting started, and the second was it can only add to a free 3' end because it's building it in the 5 to 3 direction, right? Which is what you see here. All right, so DNA polymerase 3 is over here laying down DNA nucleotides, and it's going to keep going as long as it can, which is as long as there is DNA to be copied. So notice it stops when it gets to the fork, okay? Sorry, got ahead of myself. All right, So doing pretty good on this strand, right? This is called the leading strand because notice this new piece or this new strand of nucleotides, once it got started, it just kept going. Okay, so we call this the leading strand. But we do have one problem. We have RNA nucleotides in our DNA strand. We can't have that. They're not the same thing. So we need to get rid of this RNA primer. That's where the big sister, DNA polymerase 1, comes in. DNA polymerase 1 removes that primer. Well, now we have a new problem, right? Now we have this region of DNA that's missing. So DNA polymerase 1 is going to add the correct DNA nucleotides. But notice this gap, y'all. DNA polymerase 1 has a limitation. It cannot connect what it just laid down with the existing strand of nucleotides that DNA polymerase 3 had laid down. Okay? So we've still got a little bit of a problem. We're not quite done with this strand of nucleotides yet. This is where our final key player comes in, and that is a ligase. If you've ever heard of the word ligate, ligate means to connect, and so that's exactly what ligase is going to do. It comes in and it fills in that gap. Okay? So again, we call this newly made strand the leading strand, and it was made continuously. Once DNA polymerase 3 got started, it just got busy, and it worked until it ran out of space. Okay? And it was moving, or I guess I should say it was made in the direction of that fork. Okay? Well, now we have this other strand down here, and this is called the lagging strand. And you're going to see why here in just a second. And I'm sorry, I should have scooted this up so that the captions wouldn't cover it, but that's okay. So we're going to use those same key players as last time, but it's going to be done a little bit differently. And that's because of the directionality of this bottom strand. So remember, Primase has to get the party started, and so it does. But look at the directionality. Okay, what you can't see right now is the orange strand at this right-hand end. The orange strand has the 5' end. So this piece of RNA is going to be anti-parallel. So the RNA primer has the 3' end right here and the 5' end right there. It's just anti-parallel. But this is a problem because remember, DNA polymerase 3 can only add nucleotides to the free 3' end. Well, we don't have a free 3' end, right? This 3' end is the end of the molecule, right? Or at least of that replication fork. So there's nowhere for DNA polymerase 3 to go. So to solve this problem, Primase lays down another primer a little bit farther down. Well, that's good because now we have a 3' end. So DNA polymerase 3 can hop on and fill in that gap. Well, that's good, but we still have all of this over here, right, that hasn't been created. So Primase comes back in and lays down another primer. Sorry, there we go. And then DNA polymerase 3 fills in that gap. Guess what happens next? Primase lays down another primer. DNA polymerase 3 can fill in the gap. And so this happens until it runs out of available template DNA. So now, y'all, we have a lot of RNA mixed in with the DNA. We can't have that. So who is responsible for removing that primer? Big sister DNA polymerase 1. So DNA polymerase 1 is going to come in and remove that first primer. Then, remember, now we have a gap. We can't do that. So DNA polymerase 1 then lays down the correct DNA nucleotides. Again, the limitation is that it cannot connect what it laid down to what had already previously been laid down. So ligase will come in and fix it. So now the same thing is going to happen at each of these RNA primers. All right, so I'm going to show you again. So make sure you're looking at the screen. So this red RNA primer is going to be removed by DNA polymerase 1, who is also going to lay down the correct nucleotides. then ligase will come in and close the gap. Then we go to the next one. DNA polymerase 1 removes the primer, adds nucleotides, ligase fills in the gap. Same thing at the next one. Primer removed, DNA nucleotides laid down, ligase fills in the gap, and so on and so forth. So notice this strand, which we call the lagging strand is made discontinuously. It was made in pieces, and we call those pieces Okazaki fragments, okay? Based off, it's just named off the Japanese scientist that figured out this process. So these Okazaki fragments are why the lagging strand is made discontinuously, okay? And so it's normally happening kind of away from the fork. So leading strand made continuously, lagging strand made discontinuously. All right, so here is an opportunity for you to pause the video and practice what you remember. I always recommend doing these as soon as you get to it, just to see what stuck and what didn't. Now, a lot of this is new material, so do not be discouraged if you can't correctly match any of these right now. That's okay, but just trying is helping your brain learn to retrieve this information. It's also helping your brain process the information, because when you come back and you say, okay, ligase does this, then again, it's helping your brain learn. So then you can come back after you study a little bit and do it again and see how much more you remember. So remember, I'm going to go in order here. So remember, ligase, it gets its name because it ligates or connects pieces of nucleotides together. So ligase is responsible for connecting those Okazaki fragments together. If we're talking about the lagging strand. Topoisomerase, its role is to stay where the DNA is closed, because it is trying to prevent the DNA from getting too supercoiled. So the correct answer for that one is F. The single-stranded binding proteins, that's what SSBP stands for. The single-stranded binding proteins are going to prevent the parent strands from going back together once they've been separated. So that is going to be E. They keep the parent strands separate so that the cell can start creating that new strand of nucleotides. All right, DNA polymerase 3, the captions are actually covering the answer here. Remember, DNA polymerase 3 is the main player. It lays down most of the DNA nucleotides. So it is responsible for creating that new strand of nucleotides. Remember, helicase really gets the entire process kicked off, because helicase is the enzyme that is going to unzip the double-stranded helix. It's going to break those hydrogen bonds that are keeping the parent strands together and allow them to separate. Primase is going to help DNA polymerase 3 do its job, and it does that by laying down an RNA primer. Oh, I'm sorry, that should say, oh no, that's right, okay, I was reading A incorrectly. So primase lays down a few nucleotides of RNA, which allows DNA polymerase to kind of latch on. It gives it a starting point, and then DNA polymerase 3 can do what it's supposed to do, which is lay down DNA nucleotides. All right, that leaves number 7, which is DNA polymerase 1. Remember, that's the big sister who has 2 important roles in this process. The first is going to be to remove that RNA primer that primase laid down. But we can't just have these big gaps in our DNA. So DNA polymerase 1 also fills in that gap by replacing the RNA nucleotides with DNA nucleotides. And then remember, after that is when our friend ligase would come in. All right, let's just kind of wrap this up by looking at the summary slide. And so this is just putting all of those key players together. So this is the replication fork. The blue strand is going to be the parent strands. The red strand is the newly made strands, okay? And so what's going on here is you'll see helicase, this blue triangle right here, separating the two strands. And then we have the single-stranded binding proteins latched on here, keeping those two strands apart. We've got topoisomerase working up here, making sure this region doesn't get wound up too tight, okay? And then you're going to have DNA polymerase 3, and you can see it on both the top strand, where it's making that leading strand, but also the bottom strand, okay? And so notice in my previous slide, I just kind of showed you how the Okazaki fragments were made, but like I mentioned in the text, it's technically made going away from the fork, so you'll see that here, how the arrows are going. I really was just trying to stress to you that it's made in pieces. So what's happening here is you can see the Okazaki fragments, okay, ligase has started to combine them together right here, and then it's kind of in the process of happening over here. And in this nice little illustration down below, you can see how ligase is going to be connecting the strands laid down by dipole 1 and the strand laid down by dipole 3. So this is quite an intense process. Okay, I highly recommend that you take some time and really try to sketch this out yourself and kind of talk it out loud. And once you've done that, see if you can summarize this process in only 3 to 4 sentences, okay? And to kind of write it out and then go back and look at the picture or listen to this video and see, did you include all of the key players? Did you correctly identify, the order that they work in and what they do specifically? And if not, no biggie, go through, edit your little paragraph, then scrap it and start all over. And do this until you can accurately write out this process from memory. All right, y'all, let me know if you have any questions.